Thank you for the introduction, sir. And it gives me great pleasure to present the usage of GROMACS as a part of the chain breads application. And I thank the organizers and the coordinators of the project to inviting me to this conference. And I'm here to present about the usage of GROMACS to understand the biomolecular interactions through molecular dynamic simulations. Actually, what is molecular dynamic simulation? It is a computational prediction method which understand the dynamic properties of the biological system. As the professor has previously introduced about the structural prediction of the protein and the functionality of the protein which acts in the biological process doesn't stand there. After the structure has been determined or the structure has been elucidated, it has to be analyzed how it functions or how the dynamical process happens to enable the protein to go from structure to its biological function. This molecular dynamic simulation helps us to understand the dynamic properties. It's also a configuration of the systems by Newton, integrating the Newton's law of motion using theoretical force field, where we'll give a virtual environment to the protein using water constraints, everything, and we'll calculate the time dependency of the molecule over a period of time scale. It generates information about the static dynamic properties of the molecules based on its atomistic interaction. So this helps us to study the structural changes happening in the protein at a molecular level. What is the main and challenge of this simulation is the time scale. All the calculation of the time scale start with the bond vibrations, which happens in femtoseconds, which is very small. So these softwares calculate the interaction from femtoseconds. But to understand the function of the proteins, these time scale has to reach till picosecond, where it will be the collective vibrations and the conformational transformations happens in nanosecond, but the enzyme catalysis happens in microseconds. So a normal 8 CPU, 8 core machine can just run a 2 nanosecond simulation in 1 hour using this GROMACS application. So to bridge this time gap for simulation, the necessity for high performance computing cluster is needed. Coming to GROMACS, GROMACS is an open source software which is a featured molecular dynamics package for the simulation of protein, lipids, and nucleic acid. It can run almost on all Linux-based platforms, and it has its own GROMOS package, which is a theoretical force field applied by this application. And it has a better advantage over other similar simulation packages because it has a variety of force field options, and it also has various built-in pre-processing and post-processing tools to flexibilize the analysis of these trajectory files, and also recent implementation of CUDA-based GPU acceleration is available in the recent versions. This software can be compiled either with the standard MPI protocol, or even they have their internal own threaded MPI. GROMACS has strong focus on efficient parallelization from multi-threaded to multi-level and multi-CPU, and even hybrid acceleration. The challenge is coming to the time scale. The GROMACS are better over the other commercial molecular dynamics packages such as Amber, Charm, and Desmond. So most of the international researchers who are running molecular dynamic simulation use GROMACS because of the flexible features and force field options. And coming to the recent versions are highly faster than the previous versions that had been used. And most of the Indian researchers has been extensively using this GROMACS for various biomolecular analysis, which can be both done in a CPU-based simulation or GPU-based simulations. But the hurdle is the proper availability of clusters for the researchers to understand the protein and the biological functions at the higher time scale level. I'll just talk about one of the case study that we had been working on, and just I'll make a short note of this case study. We have been working on development of an anti-diabetic molecule by targeting the protein tyrosine phosphatase 1b protein, which has a major role in the insulin signaling pathway by acting as a negative regulator of insulin receptor. And that is the structure of PTB1B, which is a experimental structure. And these were the two compounds which had shown better and anti-diabetic activity in the experimental cell line based assays. So we want to probe how it could be a better active compound for this, specifically this protein, using the analysis of the molecular interactions. And this simulation involves following steps. The force will has to be applied by selecting the protein structure, and the coordinates and ligand topology files should be created for the compatibility with this software. Then we add a water box around the protein, as you can see in the picture. Then we use the energy minimization techniques, which is around using algorithms such as steepest descent. Then we do a short position restraint dynamic for a 50 picosecond, where we froze the protein and allow the subsequent movement of the water molecules around and inside the protein. Then we go to the actual production MD run, which should be carried on until the protein gets stabilized. And these are the surrounding parameters, which shows that the NPT, which is the constant number of volumes, pressure, and the constant temperature, using the PME particle mesh evolved algorithm, which is a grid spacing of 0.12 nanometer, and the constant temperature that is kept for this protein environment is 300 Kelvin, and the pressure at 1 bar. The time step was used was 2 femtoseconds, and the coordinate stored for every 2 picoseconds. Totally, the production run was run for 10 nanoseconds each, and there was total of 10 simulations. The trajectory analysis was performed using the same GROMACS package along with GRACE. And this system size was for this protein is 300 amino acid protein, which is consisting of, along with the water box, consists of 75,000 elements. The total number of steps was 50 100,000, and the output of the simulation file was 8.5 GB. And the number of CPUs which were used for this each simulation was around 32 to 64 CPUs for each job. So this entire 10 nanosecond simulation was finished in around 5 days. This was a previous simulation which was run in the version of GROMACS 4.0.7. This was the version around 2010. The resources used for these simulations were the cluster that was the part of the Garuda, which is Indian grid computing facility, which bring in all the clusters. These were the results obtained from these simulations, where the root mean square division profile shows the stabilization of the protein for the particular period of time scale. And the black one represents the protein alone without any inhibitor, whereas the subsequent colored profile shows the compound 2, which is the reference inhibitor or the control one, and the blue one represents the protein with chlorogenic acid, and the purple one represents the protein with chichoric acid. And the root mean square deviation is done, analyzed to see the stabilization of the protein, which is stable over a particular time scale. Later, the root mean square fluctuation shows the fluctuation of each and individual amino acid residues. Then the distance between the deviation plot between at the active site of the protein showed that there was a restraint caused in the dynamics of the active site. And but the radius of gyration shows that this graph is constant, so that the protein does not get unfolded or disrupted. So to enable the functionality of the active site, it was proved that there is a alpha 7 helix at the allosteric site of PTP1B, which has been missing in the actual crystal structure, which was used for simulation. So using the homology modeling technique, this alpha 7 helix, which is shown in the orange color, has been modeled to the protein even after using this ligand. So after modeling the alpha 7 helix, the simulations were again carried out, which showed that the RMSD profile was same and stabilized as before, but there was a significant fluctuation observed at the active site region for the native protein alone, which was constrained and restricted for the other simulations of with complexes. So this picture shows the deviation in the active site, whereas this distant deviation plot between the As per 181 and WPD loop and 16 to 15 of active site shows that the protein alone enabled subsequent fluctuation of point 1 angstrom to point 0.3 angstrom, which was hindered in the presence of any inhibitor at the allosteric site. This shows that this dynamics happens only in the presence of alpha 7 helix, and the protein alone was able to get into the active state due to this simulation. So this enabled us to study the what is the dynamics happening at the molecular scale. Similarly, we study the interaction at the allosteric site of PTP1B by seeing the distances between these three important helices, which is forming the allosteric site of PTP1D, and restricting these helices were indirectly responsible for the restriction of this active site loop movement. This can be never be seen by any other experimental techniques because it's very critical, and it requires a series of snapshots of even milliseconds and microseconds. So this tool enables to see these dynamics and these movement of residues for the protein at the atomistic scale. So coming to the summary of this case study, PTP1B inhibition assay, which experimental studies showed non-competitive inhibition. An in vitro experiment using the diabetic cell line showed that the efficacy of these compounds were effective to maintain IRS1 phosphorylation by inhibiting PTP1B. And this simulation analysis helped us to probe the molecular interaction and to show how this protein gets activated to its closed form, signifying the role. So this study enabled us to compare the activity of CGA, which was inhibiting the protein in a similar manner to compound 2. So among these two caffeine derivatives, we found that CGA was effective in allosteric inhibiting the protein activation. So we went on to the studies of this compound using in vivo mouse models. So this work was published on 2012 in Journal of Chemical Information and Molecular Modeling. Based on these analysis, we just extended our studies to probe the few other plant-based molecules. How would they interact or could be a better inhibitor to this protein than the current models? So we also wanted to study how this allosteric inhibition mechanism is happening. So, but the current scenario is that the version of GROMACS get upgraded. So it is not possible to run continue or extend the previous studies. Since the version has been upgraded, there was more effective improvements done in the application. And the time scale in the recent research stages range from in scale of 100 nanoseconds. That is a recent study which has been published in 2014 on the same protein. They went on to 100 nanoseconds, which is very difficult to perform in for us to do. So the time taken for the simulation is still the greatest challenge, and recent support from chain reads by enabling the GROMACS application has helped us to do a better time scale. So we currently continuing the part of the work, we extended those previous simulation, or we ran the protein in the recent version, which showed that after 10 nanoseconds, that longer stabilization. The first picture, which was showing we ran it for till 50 nanoseconds, the graph keeps on increasing, which shows that the protein is not stabilized, local stabilization. So we adjusted the parameters, and we ran a few other simulations in chain reads resources, which showed that the protein dynamics was very, very different from the previous observed protein dynamics. So this is what happening in the field of simulation, where researchers were trying to probe the effectively on what is the actual fluctuation is happening on. So these are the current results which we are doing and continuing the previous analysis. I would thank my guide and professor, Dr. B.S. Lakshmi, of Department of Biotechnology University. I also thank my HOD and Director Gautam. I thank Satyamani and Ravindra Reddy for the IMSC cluster facility for the 1 month which they gave for the previous simulation. Then computing facility of CDAC. I thank Dr. Subrata sir, Ms. Janaki ma'am, and Pawan and Divya. I also thank the CSR government of India, who is funding me for the PhD financial assistance. I sincerely deliver my thanks to the coordinators of the Chaintrust project and organizers of this Chaintrust conference for granting me the opportunity to share our research analysis, how we do, how we use the resources, and I thank them for for the mobility grant and this opportunity. Thank you very much.
Yeah, as a biologist, I'm thrilled to find out about these tools, but I have to be honest, I'm a bit lost here. Could you please clarify how exactly ChainReds helped out, supported this project? Did ChainRed support the development of the tool? Did it increase the buy-in of the user communities? Do you have some statistics on that? Because I'm totally lost. Sorry.
Actually, this was work on the previous analysis. ChainRed is supporting us by enabling or sharing the high performance computing resources through the GROMACS application, which they have made it available in this thing. And this simulation is similar to submitting it. Due to lack of time, I didn't show the demo, but I have it in the poster. The application for the simulation is exact like they showed it for Abinit simulations. We can ChainRed is sharing the computing resources, the cluster resources for these simulations by adding this GROMACS applications in the life sciences module.
If I may make a comment, this is a similar case to the Abinit case. As I just told you in the introduction, ChainRed has provided a new MPI-enabled version of GROMACS to this consortium of researchers. Also, he has provided a portlet which make use of this MPI-enabled GROMACS version and has facilitated the access to the different infrastructure that are being supported by ChainReds. So he has mentioned the CDAC1, which is the Indian one, which also is integrated in the testbed of the project. So it is a similar case as in the Abinit, but in order not to be iterative, we have not provided the same demo as in the Abinit and so on, so he has focused on the results.
Do we have an indication of how this helped the deployment of the tool, the user? Do we have more users using that is it about the accessibility, is it about the computer resources?
There has been an increase in the number of resources that has been used. There have been some training activities about the use of GROMACS MPI-enabled version in India, in different institutes. And there are some statistics that I don't know in my mind by now, but about the number of new trained researchers in the use of GROMACS via the Science Gateway. Okay, so if there are no more questions.